Our last category of organic macromolecules is lipids, the topic of this presentation. Your study guide asks you to state at least 3 functions of neutral lipids in the body. Neutral lipids are triglycerides. It's not important that you list 3 functions. I've got 2 functions of triglycerides listed here. Triglycerides are lipids that store energy for us because they're very large and there's so many carbon-hydrogen bonds. Another function of triglycerides is to help us absorb certain vitamins. Another important function of lipids, not related to triglycerides, or neutral lipids as they're called, is actually performed by phospholipids. These phospholipids are structurally similar to triglycerides, but of course they've got a phosphate group. They also have a nitrogen, but they're important because they form all of our cell membranes. So really phospholipids are probably the most important of all the lipids. There are other lipids that are important for cell-to-cell signaling. These include prostaglandins and steroids. An example of cell-to-cell signaling would be steroids functioning as hormones, which travel in the bloodstream from wherever they're made, either the ovary, the testis, or the adrenal gland, eventually to their target organs. Some functions of steroids would involve the growth of a tissue or an organ, development of the reproductive system and organs, and of course reproduction. Now, not all lipids have a similar structure. In actuality, this is the hardest group to develop any kind of generalization about how they look or how they're structured. However, all lipids are similar in function in the sense that they're hydrophobic by definition. Now, it is true that phospholipids do have a region that's hydrophilic, meaning a part that likes water, but much of their structure doesn't like water and is hydrophobic. These hydrophobic nonpolar regions are due to numerous carbon-hydrogen bonds, and that'll be true no matter which lipid we're looking at. So we're going to look at a few examples of lipid structure, starting with the most straightforward one called a triglyceride or a neutral fat, neutral lipid. Neutral just refers to the fact that a backbone of glycerol is bound to 3 fatty acids. We consider a triglyceride to be a polymer because there are 3 repeating units. These are fatty acids. Now, the fatty acids may vary. They won't all exactly be the same. They'll have different numbers of carbons and different numbers of double bonds between carbons, but they're all considered fatty acids because they contain long chains of carbon with a lot of hydrogens. Triglycerides are synthesized just like any organic polymer through the process called dehydration synthesis. But in this case, what happens is each fatty acid, the 3 fatty acids each, will lose a hydrogen, and glycerol will lose 3 hydroxyl groups to bind those 3 fatty acids to the glycerol backbone. Therefore, 3 water molecules are made and are byproducts of attaching the fatty acids to the glycerol backbone. So fatty acids, we could say, are monomers. They're carbon chains consisting of 4 to 24 carbons. Most fatty acids we can synthesize. We eat carbohydrates, comes in our diet, and then our bodies transforms the carbohydrates into fatty acids. However, some fatty acids are called essential. This term essential is used in nutrition to signify when something needs to be ingested. So we need to eat certain fatty acids. Linoleic acid, linolenic acid, and arachidonic acid are essential fatty acids. Now, a fatty acid, as I mentioned, fatty acids vary, but a fatty acid that's called saturated doesn't contain any double bonds between the carbons. So it's called saturated because the carbons are bound to as many hydrogens as possible. That makes the fatty acid very linear, kind of like the three can be stacked upon each other. Fatty acids that have at least one double bond are called unsaturated. And these fatty acids aren't nearly as linear as those saturated ones were, and so they get kinks in them. And the result is oil. So lipids can either be solid or liquid at room temperature. The lipids that are solid at room temperature we call fats, and they contain mostly saturated fatty acids. The lipids that we call oils are liquid at room temperature, and they contain mainly unsaturated fatty acids. You've probably heard these terms saturated, unsaturated before, maybe even monounsaturated and polyunsaturated fats. These terms refer to how many carbon-to-carbon double bonds exist within a single fatty acid. Monounsaturated fats have just one double bond. Polyunsaturated fatty acids have more than two carbon-carbon double bonds. Then you might have also heard of omega-3 fatty acids. These are interesting. They're typically polyunsaturated, but there's a double bond that exists between carbon number 3 and carbon number 4 right here. Carbon number 3 is the omega position. It goes alpha, beta, omega. So that's where omega comes from. Studies have shown that eating omega-3 fatty acids is very beneficial to our health in terms of reducing heart disease and promoting optimal brain function. You can get these fatty acids through sources such as fish, flaxseed, walnuts, and leafy green vegetables. So, omega-3 fatty acids are better for you than even unsaturated fatty acids. Saturated fatty acids are the highest in calorie content, so the unhealthiest. But there is one problem with unsaturated fatty acids, but it comes from an unnatural process that occurs in the food industry. This process is called hydrogenation. During this process, a food laboratory can turn a liquid unsaturated fatty acid into a solid saturated fatty acid. And they want to do this because it helps with shelf life, packaging, and shipping. Now that we have plastic, it's not as big of a deal, but it used to be a much bigger deal. Anyway, the process involves bombarding an unsaturated fatty acid like you see on the top here. This is an unsaturated fatty acid. The red circle shows you the double bond. And if they bombard under certain conditions this fatty acid with hydrogen, that double bond disappears, and in its place there'll be a single bond, which I can't draw small enough. Okay, now that's fine. Okay, so now you have a saturated fatty acid, easier for storage, easier for shipment, just higher in calories. But the biggest problem is that this process is not dependable. Not only does that mean it doesn't always occur with 100% efficiency, it also can revert and cause some really bad byproducts. And these are called trans fats. So what can happen is that, I'll go back to this slide, is that the process can happen in reverse, and they might not even be aware of it in the laboratory. So you get a nice saturated fatty acid, but then, oh, it reverts back to an unsaturated fatty acid, and that double bond reappears. Maybe that's not the end of the world, just for shipping and shelf life. However, when the double bond reforms, there's two different configurations that the fatty acid can develop into. One configuration is called cis, and that means on either side of the double bond the molecule points in the same direction. The other configuration that can happen is that the two ends of the molecule on either side of the double bond can go in opposite directions. That's called trans. So cis fats don't seem to be so terrible, just like any fat, I guess. But trans fats, or trans fatty acids, are not easily digested. So they tend to stay, they're not broken down. So they tend to stay in our bloodstream, number one. Number two, they affect how other fats are broken down and generally raise the level of triglycerides in our bloodstream. So the higher level of triglycerides can cause fatty deposits in your blood vessels, leading to arteriosclerosis, hardening of the arteries, plaques, blockages, and it increases the risk of heart disease. So trans fats are terrible, and if you compare all the fatty acids we just talked about, omega-3s are the best, unsaturated next, saturated fats, even though they're high in calories, are much better than trans fats. Just don't eat them. It's the best advice I can give you. Okay, another kind of lipid is a phospholipid. They are structurally somewhat similar to triglycerides, and their function is vital to the functioning of all of our cells because they make up cell membranes. They actually form our cell membranes. So phospholipids are similar in structure, except for one fatty acid has this phosphate group and a nitrogen, and it makes it polar. There's a positive side and a negative side on this fatty acid. I'll show you a picture in a second. So we're going to have a structure that's similar to a triglyceride. Triglycerides remind me of the letter E. And two of the fatty acids are what we would just say nonpolar, right? They're hydrophobic. They don't like water. But the last fatty acid has a charge on either end, opposing charges. So this is polar. So what we end up with is a structure that has a nonpolar region and a polar region. Part of it hates water and part of it likes water. Kind of a split personality, I guess you'd call it. So we're going to look at that structure now. It's a highly schematic diagram, but what it shows you is the glycerol backbone of 3 carbons. And here's one fatty acid that's normal. Here's another normal fatty acid. But the last fatty acid, notice there's a phosphate group and there's a nitrogen. So those are charged up, opposing charges. So we say that this fatty acid is polar because it has those opposing charges. This makes it unique in its interaction with water. Now, before I talk about how it works with water, let me show you some of the ways that phospholipids are depicted in textbooks. The most common is the ball and stick structure on the right. The ball represents that charged polar fatty acid. These tails are nonpolar with no charge. So this side likes water. Happy days when it's next to water. That's supposed to be H2O. The tails do not like water. All right, so there's a hydrophobic region and a hydrophilic region. We call the polar region usually a head, and those sticks we call tails. And in terms of solubility, the head tries to go near water and the tails try to avoid water. So in our bodies, this characteristic of phospholipids makes them align so that they're double layered, because we have water in our cytoplasm inside all of our cells, and we also have water or an aqueous environment around our cells. So there's water. I should have used blue for water. Oops. Sorry. So phospholipids orient themselves so that those hydrophobic tails face towards each other and away from water in the cytoplasm or in the extracellular fluid. It's pretty cool. Okay, another type of lipid is called a prostaglandin. You will never have to recognize this particular structure at all. Maybe just know the word prostaglandin and that it's a lipid. It's an important communication molecule. It's involved in contraction of the uterus, blood clotting, inflammation. It was first discovered in the prostate gland, actually, so it's involved with pain signaling as well. It's a little vague. It does so many different things, these prostaglandins. But more important is the category of lipids called steroids. All steroids are actually derived from cholesterol that we eat. So we need to have some cholesterol in our bodies in order to make estrogen, testosterone, and vitamin D. Those are all very important steroids in our body. This is the structure of cholesterol. It's a steroid, and what is significant about steroid structure is that there are always 4 carbon rings, 4 rings made up of carbons, and they're fused. So they're fused together. So notice that testosterone here also has 4 carbon rings, just colored blue instead of yellowish. So there's a lot of different functions of steroids. Let's take cholesterol for starters. That's a component of our cell membranes, and it helps with the fluidity of the membrane. Estrogen and testosterone, I'm sure you're familiar with. They're involved with maturation of the reproductive system, but they're also needed for reproduction in the sense that they help make egg and sperm. Vitamin D is also a steroid. It's actually called calcitriol, but it's involved with calcium homeostasis. So that means the level of calcium in our blood and in our bones. I wanted to mention one thing about cholesterol before we finish this unit. There's a lot in the press, as well as when you go to the doctor, about good cholesterol versus bad cholesterol. Good versus bad. Just so you know, there is only one type of cholesterol, and this is its structure. So it's not the cholesterol in and of itself that's good or bad. Instead, it's more good or bad is really related to how the cholesterol is packaged in the liver, which depends on how many lipids in general we have in our bloodstream. So the way that the liver packages cholesterol is that it puts it in these what we call lipoprotein structures. Lipo for lipid and protein for protein, obviously. So if you have something that doesn't weigh very much, you say it's low density. And generally lipids don't weigh very much. So low density lipid, that goes together. That means you have a lot of lipid in your blood. And therefore, if you have high LDL, that's bad. If instead you package lipids and cholesterol together in a structure that's what we call high density, now it typically weighs more. That means there's more protein and less lipid. That's good. That means there are less lipids in your bloodstream. So it's not cholesterol by itself that's good or bad. It's how much lipid you have in your bloodstream and how the liver can package the cholesterol. That is the end of organic chemistry. Thank you very much.